Hello, everyone. Thank you for joining us today. My name is Noel Lin, and I am a managing editor here at the American Society for Microbiology, or ASM. ASM publishes the journal M Systems, which is hosting today's webinar, as well as all others in what we call the Thinking series. For more information on this series and to watch previous episodes, please check out the URL you see on the slide here or scan the QR code. Now, our webinar today is the second part of the two-part series on critical concepts in dormancy. Part one was held in September of last year. If you attended that webinar, welcome back. If you missed it, then I invite you to visit the Thinking Series webpage to view the recording. Next slide, please. If you're not familiar with M Systems, this journal publishes preeminent work in microbial systems biology. We welcome papers on both basic and applied research and that focus on cellular systems, ecosystems, and everything in between. The journal's scope includes research on the microbiome, omics, bioinformatics, computational microbiology, high-throughput analyses, cutting-edge data tools, and a whole lot more. M Systems has a prestigious international board of editors, and we publish papers from authors around the world. We're committed to fair and rigorous peer review and an author-friendly approach to publishing. Next slide, please. I want to especially mention our recently launched call for papers on microbial dormancy. Articles that are accepted as part of this call will be featured in a special online collection. And we're looking for papers that address advances in our understanding of microbial dormancy and its consequences on health and environmental systems. We especially welcome multidisciplinary and innovative approaches to understanding the drivers and consequences of dormancy in microbial systems. And for more details, please visit our webpage at the URL and QR code you see on the screen. Next slide, please. Now, both the microbial dormancy call for papers and this two-part webinar on the same topic are spearheaded by Dr. Ashley Shade, who is our moderator today. Dr. Shade is at the French National Center for Scientific Research in the University of Lyon. The focus of her research is to understand, predict, and manage microbial communities to achieve resilience to climate change and other environmental disturbances. Ashley is currently a senior editor at M Systems, but will become, on July 1st, our new editor-in-chief. She will take over for our founding editor-in-chief, Dr. Jack Gilbert, and continue to build on a great foundation that he has established. And in addition to Dr. Shade, we have 4 internationally renowned speakers today for our webinar. Now, before I turn things over to them, let me just quickly say that if you have any questions today for our panelists, please use the Q&A button that should be located at the bottom of your Zoom screen. We are not using the chat function in the webinar today, so please submit all your questions through the Q&A option. Okay, I will now invite Dr. Ashley Shade to introduce our topic and speakers today. Ashley?
Thank you, Noel, and welcome everyone to the Thinking series. This series has been in planning for over a year now, and I am so happy to join you here today for our part 2, which is all about discriminating microbial states of activity. And I'd like to start off by thanking our panelists for sharing their expertise today and the ASM staff for their super support in organizing, as always. And also, I want to thank all of you for taking the time to join our discussion today, which I hope you will find as fascinating as I will. So let's start off with a little introduction. We know that many cells can persist in a reversible state of reduced metabolic activity called dormancy. Dormancy allows cells to survive unfavorable conditions, disperse and immigrate widely, broaden community diversity, extend lifespan, and preserve genetic information for future generations. Understanding the consequences of dormancy has wide implication across biological systems and the potential to contribute to addressing grand challenges facing humanity, from controlling diseases to facing the climate crisis. So the objective of part 2 of this series is to bring together international experts who think about the mechanics of how we assess, quantify, and distinguish states of microbial activity and dormancy. Determining how active or inactive a microbial cell, population, or community is technically and often practically quite challenging, especially in situ. And furthermore, different methods for measuring activity offer different data types that are not necessarily comparable, leading to challenges in states and ranges of microbial activities across studies. However, these data are critical for understanding the consequences of microbial activity and dormancy for microbial systems. And so the topics that we have today have a wide breadth, but we have some international experts who are going to bring you varied perspectives on these discriminations of activities. So I'll introduce our first speaker. We welcome Dr. Catherine LaRose, a microbial ecologist and biogeochemist from the CNRS Institute of Geosciences and the Environment, France. She performs research in Arctic systems and specifically is known for her contributions to assessing microbial activities and their functional contributions in snow and ice environments. These cold environments present a particular challenge for assessing microbial activities due to the relatively low microbial biomass within ice and snow communities, along with the low temperatures that permit relatively slow cellular processes. Thank you so much for being here, Catherine, and the floor is yours.
Hi, everyone. Thanks for that really great introduction, Ashley, and for the definition on dormancy, and also by providing some context for some of the environments that I study. Just before I launch into it, can everyone see my slides? Okay, so I guess I'll go. So today I'm going to be talking to you about dormancy in frozen environments. And I'm going to be focusing on snow, even though we also work on ice and the atmosphere as well. And I'm going to go through some examples and talk a little bit about how we look at it and how we think about dormancy and ways of measuring it. And as Ashley said, it's difficult because when we think about dormancy, it's the lack of activity, but the tool we can use is measuring activity. I'll just give some examples and talk through some of our results looking at this in snowpack. So what I wanted to start with is just to briefly introduce our environment of study, one that I really love. And I'm going to be talking about seasonal snowpacks. And seasonal snowpacks are really great models for studying the limits of life, ecology, biogeography, adaptation, as well as dormancy. And the reason for this is because they form every year and then they melt out. And that means that the communities that are in these environments need to either adapt quickly or have some sort of adaptive mechanisms that allow them to persist until the conditions become more favorable. So this is a picture of a snowpack of some snow, and here we see these different layers. And these layers represent different snowfall events that have enabled the snowpack to form. Snowpacks form on sea ice, on terrestrial systems with soil, with permafrost, and also on glaciers. And what we see is that the snowfall, as it falls onto the underlying surface, brings in not only snow, but also atmospherically transported microorganisms. And these organisms, once they enter the snow, either adapt, survive, or enter into dormancy in order to be able to survive and persist. And these environments are actually great systems to look at dormancy because they are under some relatively severe physical chemical constraints. There's the low biomass, as Ashley mentioned, but also relatively extreme temperature gradients. But the surface of the snow, we can be anywhere from, depending on where we are, -20, -25, almost zero at the base of the snowpack, depending on the thickness. It is also a highly radiative substrate because the ice crystals reflect incoming solar radiation. So there is high oxidative stress in these environments. But this is quenched in the first 15 to 20 centimeters of the snowpack. So again, we have this gradient across the depth of the snowpack. They're also oligotrophic environments, and because they receive particles that are transported from all across the globe, there are often point sources of contaminants. So, for example, mercury depletion events in the spring that lead to these kind of toxic events for these microorganisms. And the whole system, in addition to having these constraints, is dynamic with rapid changes related to solar radiation, temperature, and season. So yeah, so these are unique environments to look at this stuff in, and one of the ways in which we do it at least in our lab, as we said, as Ashley mentioned, there's so many ways of looking at activity, is we base our work kind of on two systems. So field studies. This is a picture in Svalbard where we gear up, we go out and we collect snow samples. And we like to do time series sampling. So depending on the amount of time we're able to be out in the field, we'll do it for weeks or months at different frequencies, depending on the question. And then we also like doing lab experiments in microcosms, where we take our collected snow and then we incubate it in the lab under different conditions. And in these systems, we do several different approaches. So we've been using more recently metatranscriptomic and kind of metagenomic approaches that we couple to markers to look at communities. So here I have 16S, but of course we look at 18S as well. And then we also try to link this to chemistry. And because it's so difficult to measure dormancy and activity, we try to use a bunch of different tools and see whether and how well they fit together. And then when we do our lab studies, we also look at different types of methods to assess activity, including respiration, and then we can follow metabolic transformations. And so illustrate what we've been seeing in the snow, I'm going to go through an example of a field study where we are looking at metagenomics and metatranscriptomics of a snowpack that has been sampled year-round in Svalbard. So I'll get to that in a minute. So what we see is that in these snowpacks, so here we have the community structure over time. And what we see is that we really see a strong seasonal shift. So the winter snow is very similar in terms of how the communities are structured. But then if we look towards the spring, where in the Arctic, the light is coming back, we have a different kind of community, or at least community structure, in March, April, and May. And we see that these communities shift relatively quickly over short periods of time. And so one of the things we were interested in trying to understand is how these communities are changing and what are they doing and what's driving these shifts in these communities. And the way we've been doing that is looking at function and functional activity. So in this case, we zoomed in on April and May, and we collected samples every 3 days for metagenomics and metatranscriptomics. And then we also did some daily sampling for microbial community composition. But for the purpose of today's talk, I'm going to really focus on the functional analysis. And one of the ways we thought of estimating activity, or at least shifts and differences between these communities, is to use overlap analysis to look at the difference between protein sequences in the DNA versus the RNA data set. And then use what's being transcribed to identify or help try to identify drivers of selection and what's really driving the activity or dormancy of these communities. So another way to show this is by looking at the overlap again, as I mentioned. So these are protein sequences. This is our early spring data from April. We see that there's a low diversity of protein sequences that we found in our system. And then what's really interesting is if we look at the mRNA, so the metatranscriptomes extracted from these same samples, we see that only a small portion can be mapped back to the metagenome. So this would suggest that only a small portion of the organisms in our DNA, in our community, in the DNA set, are active that are dominant, both dominant and active in our early spring. And we also see that we have a relatively large proportion of these kind of phantom sequences that are in the transcriptional data that we're unable to map back to the metagenomes. So this is probably a mix of insufficient sequencing depth of the community in terms of the metagenomics, but also that these rare taxa are the ones that are dominantly active in this early spring season. And then what we see is then when we look at what's active in the early spring and then what becomes dominant in the microbial community, we see that there's almost an 80% overlap. So it means that this small fraction of rare organisms that are active in the early spring end up becoming dominant in the late spring samples, so in our May samples. And what we also see is there's a much stronger functional overlap in this late spring samples between the DNA and the mRNA. So a higher fraction of the community is active, so represented both in the metatranscriptomic and the genomic data sets. And so we're seeing this shift in dormancy and activity. And we're doing this, as I said, with metatranscriptomics and metagenomics. But a recent study published by James Bradley from the Bradley Lab in Marseille looked at this kind of shift in activity on glacier snow and ice and came to relatively similar results. So what he's been observing is consistent with measurements for snow on glaciers. And he used BondCat. So in this sense, he used protein expression and not necessarily metatranscriptomics to look at activity, although he did use metatranscriptomics as well. And he falls within the same range. So in his melting snow, he sees activity between 50 and 65%. And in the snow, using a different method, we're at about 71%. So it looks like even though the numbers aren't exactly the same and it's not the same environments exactly, we're looking at environments where there's water have more of an overlap between active and inactive cells. And so one of the reasons metatranscriptomics is also actually really cool is we can look at the functions being expressed and transcribed, and then we can see whether these can give us some hints about what the communities are facing in terms of their lifestyle and activity. So if we look at what they're doing, we see more genes that are transcribed in the metatranscriptomics set in this early April sample related to water acquisition. So a lack of liquid water. We're seeing carbon starvation. We're seeing genes related to nitrogen starvation, so nutrient starvation. But then we're also seeing genes related to genetic modification, so adaptation. And then when we look over to what we're seeing in May, we see genes not related to nutrient starvation anymore, but more related to community interaction. So competition, virulence, antibiotic production. But we're seeing this strong shift in what the communities are doing. And we think that this can help us understand what's driving maybe some of the changes in activity that we're observing. So how do we do that? So to do this, we went back to the environmental and geochemical data. And what happened in April? So a couple of things happened in this April to May transition. We increased the temperature in the snow. So we went from an average of minus 15 degrees Celsius to minus 5. So we're looking at more or less a 10-degree temperature increase. And what we also observe in these Arctic systems, so our sampling site was close to an open fjord. And we saw that in that first week of May that we had an algal bloom in the fjord, which led to a 4-fold increase in organic carbon that we could measure in the snowpack. So going from 10 ppb to 40 ppb. So we completely shifted the nutrient levels and also the temperature over this time. And so if we go back to if we're thinking about drivers, we could think about things like liquid water, because as we increase the temperature, not only does it get warmer, but we increase the amount of water in the system, and also this increase in nutrients. And what this means is if we go back to our question about dormancy, we can look in our early spring DNA set where we have a certain amount of dormant organism and see to what extent they're represented in the DNA and RNA data sets in the late spring. And we were able to see that by shifting the temperature in the water and the nutrient availability, we've rescued roughly 50% of the functions that weren't observed in the mRNA data set. So these environmental changes do have an impact on dormancy and activity across temperatures and seasons. And so to bring it back to what this means in terms of climate change, well, there's a few things we know. So the predicted changes or the expected changes are increased winter warming, So we're expecting an annual temperature rise of 4 degrees over the next few years, or it has experienced that. And during the wintertime, we've observed a 7-degree increase between 1971 and 2017. Based on our field results, we can show that sub-zero warming, so even staying below zero, will increase both microbial respiration and activity, and will likely reduce dormancy, which will have an impact on climate gas emissions as well as biogeochemical cycling. We know there's going to be an increase in rain on snow events, and the consequence of this is adding liquid water into the snowpack. As we've seen from this example that I showed you, but also other studies, liquid water or adding liquid water into these frozen environments will increase activity and growth. But in terms of the dynamics of the system, if this water freezes, it might also impact microbial survival. So it means we're likely going to reduce ecosystem predictability, which could change the types and the range of dormancy that we see in these communities. And then another predicted change or expected change is an increase in aerosol emissions, with increased deposition of chemicals, particles, nutrients, and also microorganisms, which will change the microbial communities in snowpacks. And as we've seen, more carbon will likely reduce dormancy, but if we increase pollutants like mercury or lead or antibiotics, this might have an impact on microbial survival, and again, leading to more ecosystem instability and unpredictability. So I think I'm going to leave it at that. And just before I sign off, I would like to thank all the funding agencies. So doing polar field research is extremely difficult, and we depend on a lot of collaborators and institutions to help us out. So I'd like to acknowledge them. I'd like to acknowledge the former members of our lab and then the new members of our lab. And then I'd just like to shout out the Bradley Lab at Université Aix-Marseille, because James has been doing quite a lot of work on developing new methods to study and model dormancy. So thank you very much.
Great. Thank you so much, Catherine, for that great kickoff to our webinar. And also, I love how you were showing the temporal component of looking at those dynamics of activities and the contribution of one time point to the next. I think it's really great that you're able to glean those insights from your system, and I hope we can talk more about it at the end. If you have questions for Catherine, please feel free to put them in the Q&A box. She might be able to answer some of them like in the box, and we might bring some up for discussion at the end. So please hold on to those questions, and we look forward to them. All right, I'd like to move forward and introduce our next speaker. So for our second speaker, we welcome Professor Yingying Pu, a molecular microbiologist at Peking University in China. She is known for her contribution to understanding the molecular mechanisms that define persister cells, which are subpopulations of bacteria that can withstand exposure to antibiotics. She has brought insights into the concept of dormancy depth, which is thinking about how persistent a dormant state might be. And she's demonstrated it using assessments of protein aggregations within a cell, and she's going to share that with us today. So thank you so much for joining us, Yingying, and the floor is yours.
Thank you. Thank you for the great introduction. Let me start. Our lab has been deeply engaged in the study of persistent cells. Persistent cells is a phenotypic antibiotic tolerance and have significant clinical implications. Persistent cells are closely related to dormant states of the cells. So I'm truly honored to have this opportunity to share our latest findings with you today. My topic is modulating microbial latency. Functional crosstalk between bacterial aggregations and the cellular dormancy. Let's start with a fundamental question. What is dormancy in microbes? Dormancy is a state where bacteria enter a non-replicative, metabolic inactive state to survive environmental stress. This ability is crucial for their survival, especially under harsh conditions such as antibiotic exposure. So why is dormancy important in clinic? Well, dormancy is a major cause of antibiotic tolerance and chronic infections. Understanding the mechanisms behind dormancy can lead to new strategies to combat antibiotic resistance, which is growing global health concern. Sorry. One of the most intriguing states is that of persist cells in dormancy, which survive antibiotic treatment but have the potential to resuscitate after antibiotic removal. And this is in contrast to available but non-culturable cells, which is Weber's cells. Weber's is in a deeper state of dormancy and often fail to resuscitate even after the antibiotic is removed. The biphasic killing curve is considered the gold standard for measuring persist cells. It shows two distinct phases. The first phase where most cells are cured, and the second phase where a small subpopulation of cells survives. We found that if we prolonged the duration of stress treatment, the persistence ratio actually increased. This suggests that the duration of stress exposure plays a critical role in determining the depth of dormancy and the likelihood of cells surviving antibiotic treatment. To further investigate this phenomenon, we monitored the survival dynamics of these cells under real-time microscopy. Here's what we observed. Most cells last under antibiotic pressure, but a small subpopulation of dormant cells survived for at least 6 hours. When we removed the antibiotic and allowed the cells to recover, we identified two distinct types of persist cells. The first one is fast recovery type. These cells resume growth within 5 hours. They represent the classic persist phenotype. And the second group is the slow recovery type. These cells took between 12 and 48 hours to resume regrowth. This is a novel intermediate state we identified. And the last one, the Weber's cells, this remained dormant for over 3 days. We cannot check its recovery, indicating a very deep dormancy state of these cells. Based on the observations we just discussed, we propose a dormancy depth continuum in bacterial persist cells model. Let me walk you through this model. Imagine dormancy as a spectrum. The depth of dormancy determines how long it takes for a bacterium to wake up, or what we call the lag time. At one end of the spectrum, we have shallow dormancy. These are the easy wakers, or what we call persist cells, fast recovery. They can bounce back very quickly. Moving further along the spectrum, we have intermediate dormancy. These are the delayed wakers, or persist slow recovery. They take a bit longer to recovery. So the key takeaway here is that bacterial dormancy is not just a simple on-off switch. It's a spectrum. This continuum helps us to understand that there are varying degrees of dormancy, each with its own implications for bacterial survival and recovery. When we observed those dormant cells under differential interference contrast microscopy, we noticed a distinct phenotypic feature. It's dark foci at the polar positions of the cell, around 200 nanometers in size. Initially, we were very puzzled by what these structures were, and at that time, we named them just aerosomes. To further investigate, we developed a method to purify these aggresomes from the lysed cells. We purified aggresomes exhibited. This purified aggresomes exhibited a generally spherical shape and remained intact throughout the lysis and purification processes. This discovery was crucial because it allowed us to study these structures in detail. So next, we use the mass spec to identify the protein components of the aggresomes. We found several key markers, including FPA, LON, and HSAU. If you know these proteins are typically associated with stress responses and protein aggregation, suggesting that aggresomes play a significant role in bacterial stress response. Next, we measured the frequency of agrosome formation across different states of dormancy. We found the correlation between agrosome formation frequency and the dormancy depth. This result suggests that agrosomes are not just a byproduct, but a critical component of the dormancy process. So what drives the formation of agrosomes? We explored several factors and found that agrosome formation correlates a drop in pH. However, pH alone, with modulation of pH alone, is not sufficient to induce agrosomes. This must be other factors at play. One of the most critical factors we identified is ATP. We observed significant ATP depletion in dormant cells. Our deduction is that ATP is crucial for maintaining protein solubility and preventing aggregation. Experiments confirm that ATP depletion leads to widespread protein aggregation, resulting in the formation of agrosomes, which is labeled by BPA or LON or HSAU. Further, to further validate our findings, we also used arsenite, a well-known oxidative stressor that causes ATP depletion. Our experiments showed that arsenite treatment can effectively induce cellular ATP depletion and agrosome formation. And another condition we explored was the expression of HOCB. HOCB is a toxin protein whose expression leads to ATP depletion. Both conditions consistently resulted in agrosome formation, reinforcing the link between ATP depletion and agrosome formation. To understand how dormant cell resuscitate, we conducted real-time imaging to observe the dynamics of agrosomes. Our findings revealed that agrosomes form and dissolve dynamically during antibiotic treatment, after antibiotic treatment and resuscitation. Importantly, we observed that DNA foci co-localized with the agrosomes and precede resuscitation. This suggests that the clearance of agrosome is a critical step in resetting cellular protein homeostasis and allowing cells to resume regrowth. At the heart of this process is the DNK cell BP, the two proteins. They form a protein disaggregation machine, which acts as a key facilitator of resuscitation. Specifically, ATPase. These are the mutants of ATPase deficient DNK mutants, fail to form functional foci. No DNA foci anymore, and cannot clear agrosome. And the possible explanation is that DNA binds to the protein aggregates within agrosomes and recruits cell BP. The complex then dissolves the aggregation in an ATP-dependent way, effectively restoring proteostasis and enabling the cell to resume normal function. Our mutant study revealed that DNK and cell BP mutants show increased dormancy but reduce the resuscitation rate. This highlights the critical role of ATP-dependent machinery activity in resuscitation and suggests that targeting this pathway could be a promising strategy to combat antibiotic tolerance. So, if there are any other components in the agrosome, this is the key findings of our research. Agrosomes are crucial marker of bacterial dormancy depth. ATP depletion drives agrosome formation, leading to the dormancy phenotype, and this complex are essential for resuscitation bacterial agrosomes, and agrosomes selected contain mRNAs. Agrosome can protect mRNA integrity during stress conditions and facilitate cell resuscitation after removal of stress factors. And in the future, we want to investigate how agrosome protect mRNA from long-term stability and whether agrosome and the functionality of the agrosome biology. So thank you very much, and I have to thank all those members in my lab. They are very hardworking for the project, and my collaborators, Mark from University of York in the UK, and Professor Fanbai from Peking University. Thank you very much, and also my fundings. Thank you.
Thank you so much, Xingying. What an interesting presentation, really digging into the different adaptations and cellular mechanisms supporting a dormant state. Please put your questions for Ying into the Q&A. I'm sure you have a ton. I know I do. Super fascinating stuff. We're going to move on to our next speaker. For our third speaker, we welcome Professor Rachel McElprong, an environmental microbiologist with expertise in genomics working at California State University, Northridge, United States. She studies the activities of microbial populations that live or can live in harsh environments and environments in transition due to climate change. This includes thawing, permafrost, and even, yes, outer space. She's going to be sharing some of the ways she thinks about measuring activity in these challenging environments. Thank you so much for contributing, Rachel, and the floor is yours.
Thank you very much for the generous introduction. Unfortunately, I'm not going to be able to talk much about outer space today, but I will be talking about some environments that are almost as neat, and that is permafrost. So to kick this off, let's explore a brief introduction to permafrost, and specifically what permafrost is. The formal definition of permafrost is permanently frozen ground that's been frozen continuously for 2 or more years, but most permafrost is much older, and it's distributed widely across the Arctic and subarctic, and it's typically overlain by the active layer, which freezes and thaws annually. And microbial life persists in permafrost, and what you're seeing here is an image taken by my collaborator, Mark Waldrop, of permafrost, which shows some of the brine channels that form. So salt depresses the freezing point of water, providing a liquid environment, even though the permafrost is below freezing. And these microorganisms can live in these liquid brine channels, though the metabolism and activity is very slow. Now, in permafrost, distinguishing between active, dead, and dormant cells is particularly challenging. And one of the reasons for that is that frozen conditions preserve detrital biomass. So when cells die, the DNA can be preserved for long periods of time. And the low activity and low biomass make RNA-based studies mostly infeasible, and there are a few exceptions to that, which I'll talk about a little bit. So we need other ways of addressing issues of microbial activity and dormancy in permafrost. And there is no one magic bullet. Instead, our lab has tried to come at this problem from a large number of different ways to try and understand the activity of microorganisms in permafrost. So the first bit of information I'm going to tell you about is the exception to the rule. And it's one of the handful of studies, maybe 2 to 3 studies, that have successfully extracted and sequenced RNA from intact permafrost. So this isn't thawed permafrost, but intact permafrost. And in this particular case, what we did is we looked at the representation of metatranscriptomic reads to metagenomic reads. And this took place at the Bonanza Creek LTER site, and 3 types of soils were sampled: active layer, bog, so that bog had formed because the underlying permafrost had thawed and the ground subsided, and then there was a bog formation, and permafrost. And this particular permafrost was very recently formed, which is what enabled the metatranscriptomic approach. And so you can see here the a diagram of the core, and the location where the permafrost was sampled was very near the transition between the permafrost and the active layer. And so this permafrost had been recently formed, and it's possible that in warm years that occasional thaw had occurred. And what you're seeing here is a ratio of relative abundance of metatranscriptomic to metagenomic information. And so an excess of metatranscriptomic reads compared to metagenomic reads indicates higher activity than you would expect based on just the abundance in the samples. And what I will point out here is members of the firmicutes phylum showed in the active layer and the very recently formed permafrost had a higher activity than would be expected based on their abundance, indicating that endospore forming taxa are active in these samples. So just because you're an endospore doesn't mean that you're dormant. So with the caveat that lots of different types of dormant persister cells can exist in permafrost, one of the first places to look is at the endospore forming taxa. And in a recent paper, we sampled a large number of different permafrost sites across the Arctic. And we performed we sequenced many metagenomes. And we extracted the 16S rRNA reads from those shotgun metagenomes, and we used those to infer taxonomy. And what you're seeing here is a distribution of the different phyla across those sites. And in the orange, which I've got circled, are firmicutes, which are, of course, the endospore forming taxa. And one thing to point out is that the relative abundance of endospore forming taxa, or firmicutes, differ enormously across permafrost sites. But we're going to focus on the Fox permafrost tunnel, which is one of my group's primary study sites. And I think this is a super neat, super cool, if you will, study site. It's run by the U.S. Army Corps of Engineers, and I work with my collaborator, Tom Douglas, quite a bit at that particular site. And it's a tunnel that was bored into the permafrost in the 1960s. So in the bottom left, you can see the tunnel portal entrance. And once you open up that somewhat unassuming door, it's a whole world of permafrost. And one of the really neat things about this particular sample site is that you can access Pleistocene age permafrost from inside the tunnel. So near the front of the tunnel, you have permafrost that formed about 19,000 years ago. So this is in substantial contrast to the very young permafrost that I discussed in the last example. And as you go further and further back into the tunnel, you can access permafrost of increasing age. And also active layer soils and Holocene age permafrost, so a few thousand years old, are accessible from above the tunnel. Now, in one of our studies, we looked at the changes in taxa and the genetic structure of microbial communities through time, using this chronosequence that we can access in the tunnel. And one of the things that we found is that the relative abundance of endospore-forming taxa increases with permafrost age. And so one of the questions that brought up is, does that mean that as permafrost increases in age, that the primary survival mechanism is a dormancy strategy? So, like I mentioned, this is really difficult to look at using standard techniques like RNA. So one of the methods that we used is this endospore enrichment protocol, where we applied a number of treatments to lyse the vegetative cells and remove DNA from those lyse cells and from the detrital biomass that was already there. So in principle, what remains after that treatment are endospores. And when you extract the DNA and you sequence it, you are sequencing material from the endospores and not the community. In reality, when we test this with, say, E. coli spike-ins, we get about an 80% reduction in E. coli cells. So it's not perfect, but it is an enrichment and can give clues as to what's going on. We also extracted and sequenced the ribosomal RNA gene from the entire community, and we can use that for comparisons. So one of the things that we did is we divided the members of the Firmicutes phylum in permafrost into the two major classes, Bacilli and Clostridia. And what we're seeing here is in the 19,000-year-old samples, you have an enrichment in bacilli in the treated samples versus the untreated samples, indicating that the bacilli are likely to persist as endospores. At the 27,000-year-old time point, you see what looks like an increase in that trend, and still there's no change in the Clostridia. And interestingly, in the 33,000-year-old samples, you have this really big difference between the bacilli and Clostridia, where the bacilli are likely to exist as endospores, but then the negative change in relative abundance suggests that members of Clostridia class are more likely to persist in a non-dormant state. And that makes sense when you think about the broad generalizations in terms of what members of these taxa can do. And in bacilli, these tend to be aerobes or facultative anaerobes. And in Clostridia, these generally have a lifestyle that are more conducive to survival in permafrost, such as being mostly obligate anaerobes. They have fermentive metabolism, and they persist on detrital biomass. So there are other ways that we can start to look at measures of microbial activity. So this is a stable isotope probing experiment that we did at sub-zero temperatures. And this was done by MC Lewis and Mark Waldrop. And they filled a cooler with 70% glycerol, fitted it with a recirculating pump, and maintained the temperature at -3 degrees Celsius for over 270 days until there was mechanical failure of the system, which was unfortunate, but we still were able to perform the experiment. So just as context, for those of you who aren't familiar with SIP, what you can do is you have your sample and you add labeled substrate to it, and you can measure the labeled carbon dioxide that comes off of it. And then you can also separate the labeled versus unlabeled DNA, and get an understanding of the communities. So these are the data showing the cumulative 13C respired, indicating respiration and processing of the labeled glucose that we added by active members of the microbial community. And it takes a very long time at sub-zero temperatures for us to start to see a result. But in all of the different time points from the younger Holocene permafrost to the more ancient Pleistocene permafrost, we did see evidence of microbial activity. And the sequencing is in progress. Like many experiments with the permafrost, getting the sequencing and other assays to work can be difficult and painful, but we should be able to have data in the nearish future. Oops. So another way that we can get at the dormancy issue is to look at measures of microbial activity. So using extracellular enzymes. So we did that. We looked at several extracellular enzymes that were capable of or involved in degrading different types of substrate that we might expect in permafrost. And we do see that there is extracellular enzyme activity in all of our time points. And what is notable is that for the extracellular enzymes that are involved in the degradation of microbial necromass, that the trends suggest that perhaps this becomes a more important carbon source, carbon or nitrogen energy source in the older samples. And you also see some evidence of the degradation of peptides as well. So we also measured ATP activity, which is ATP, which is a measure of potentially active cells. And we looked at both the intracellular and extracellular ATP. And again, the take home is that we have evidence of activity in these soils. Okay, so currently we're working with the JGI on a new method for trying to determine activity in permafrost. And we're attempting to use something called bond-cat FACS, which is where you incubate your sample with a methionine analog, and the methionine analog, HPG, is incorporated to the proteins of active cells. Now, the nice thing about this method compared to SIP is that this method doesn't require replication and DNA synthesis. This just requires that they be making proteins. So once they've incorporated the HPG, they can be fluorescently labeled and then sorted using fluorescence activated cell sorting. So this is an ongoing project, and a edge case use of this technology. And so hopefully we'll get some good, interesting information using this. So in just the last couple of minutes that I have, I want to talk a little bit about microbial activity when permafrost thaws. And we performed some mesocosm thaw experiments where we took active layer Holocene permafrost and Pleistocene permafrost. And we distributed them into these 0.2 micron nylon membrane bags, which prevents the microbial exchange with the surrounding environment. And we transplanted them into the active layer to simulate thaw. And there are some nuances to this experiment that I won't be able to get into at this point. But we have a large number of data types to try and understand what is going on with microbial activity when permafrost thaws. So just a quick overview of some of the differences. We have here the different soil types and the different time points. We've got unincubated soil, 2 weeks and 2 months. And you can see substantial differences between the soil types, and especially in the Pleistocene permafrost, between the time points. So one of the last things that I wanted to point out is the activity of microeukaryotes, because that's something that is largely been ignored. And so we performed some metatranscriptomics, because you can. When permafrost thaws, it's a little bit easier than an intact permafrost. So we took our pool of metatranscriptomic reads or rRNA, and we did rRNA depletion before sequencing of prokaryotic rRNA, which left the eukaryotic rRNA and mRNA. And we computationally identified and classified eukaryotic rRNA reads to understand who the active microeukaryotes were. And 50 to 80% of all of these were, well, 50 to 80% of all total RNA reads were eukaryotic rRNA, and a high percentage were fungal. So we have very active fungal communities in the thawing permafrost. And a couple of different types, or actually many different types, and just a few highlighted here. But I think I'm going to skip over these in the interest of time. And in conclusion, the answer is it's complicated. But we do know that they are active in permafrost. And sometimes activity is preferable to dormancy, even if dormancy is possible. But the specifics really depend on soil characteristics. Permafrost is enormously diverse. It's not a mesocosm. So it's hard to, we don't yet know the rules to allow us to extrapolate from one type of permafrost to another yet. So we need new approaches, and we have, or excuse me, multiple approaches, but we have new techniques that promise to offer substantial insights into the system. And I'll end with acknowledgements, collaborators, funding sources, and members of my group. All right, thank you very much.
Thanks so much, Rachel. What an incredible breadth of different techniques that you've shared with us today for how you try to understand who's active and who's not in your very challenging system. That's super interesting. Please put your questions for Rachel in the Q&A. We're going to come back to some of those techniques in the discussion. In the meantime, I'd like to move on to welcome our final speaker, Professor Namiko Matari from the Niels Bohr Institute at the University of Copenhagen in Denmark. A theoretical physicist by training, she became interested in applying physical dynamics approaches to understanding biological systems, so we're lucky. She studies how stochasticity and determinism differently contribute to transitions into and out of dormancy, and is going to share her expertise and perspectives in understanding those different processes today. So Namiko, thank you so much for joining our webinar, and the floor is yours.
Thank you very much, and thank you for the introduction. So I'm a physicist, and I'm mainly modeler, but I got interested in this bacterial persistence phenomenon some time ago, and I slowly also expanded my knowledge into the bacterial dormancy, and also we had also some experience in the doing experiment. And today I was asked to discuss about the deterministic versus stochastic dynamics in dormancy, and I try to provide maybe some starting points to discuss this topic. So we have already seen this the kill curve for the bacterial persistence, and the in the Yin's talk, and this sort of the slower part of the growing. The killing curve is basically indicator of the persisters. They are the subpopulation of the bacteria that are dying the slower, and though they can regrow, and then after they regrow, then they are antibiotics sensitive again. And of course, the persistence is not necessarily all dormant cells, but there have been observed many correlations. So that for the simplicity within this talk, let me sort of mix them up so to call them as kind of dormant population. So if they're non-growing, then actually this killing curve is a very interesting information, because it shows that when they are waking up, because once they start to regrow, then they will die if the dormancy is necessary for the persistence. So assuming that if we actually see this pure biphasic curve where the second slower part of the curve is the straight line in this the semi-log scale of the population, the alive population versus time, then the straight line of the slower dying population means that they are actually dying at a constant rate. And the simplest interpretation of the constant rate death would be that there is a one well-defined dormant state, and then they are just coming out when the noise kicks out. So this rate doesn't really change over time. There's no aging of this dormancy, and they just come out at a constant rate. And in other words, if you see the more complex time scales in this killing curve, then it shows the complexity of this dynamics. The killing curve does not talk too much about how the bacteria can enter the persistence, but for that there are also, of course, many different discussions. But especially interesting distinction is the difference between the so-called spontaneous persistence and triggered persistence. So the spontaneous persistence is the persisters that found in the population that are in the exponential steady state growth. So that this is clearly some kind of heterogeneity in the phenotypes within the constant environment, because the majority of the cells are also, of course, exponentially growing, and then some of them just becomes dormant, so that it have to be some stochasticity event-driven phenomenon. On the other hand, if there is stress, then that are known to increase the amount of persistent cells. So they are also called triggered persistence. And this is kind of of course, when there is stress, then entire population would somewhat at least respond to the stress. And that also affect this dormant population. So the triggered persistence have some deterministic component in the formation of the dormant cells. But then after I said this, I also want to mention that even for the spontaneous persistence, the exponential growth condition can vary. So if you grow them in, say, less rich media, maybe cells can be a bit more stressed than the rich media growing cells, and then maybe there can be the difference in the amount or the type of the spontaneous persister in the different city growth rate. And if there is such kind of the environmental dependence, then it can be also there is some kind of deterministic average behavior that is changing depending on the growth condition, and that can also affect the bacterial dormancy. Now, if we actually look at the bacterial killing curve, then for example, the spontaneous persistence, then we see the many different maybe if we do that in the short time, then maybe the biphasic curve saying that second part is a straight line may fit well. But if you actually measure relatively long time in this the killing curve, then very often the people had observed the different time scale in killing curve. So that showed that the just pure bistable picture may not be enough to understand this phenomenon. And here what we are showing is one of the data done by at that time our PhD student Mikael, and he did this killing curve for 7 days, and then we see sort of progressively sort of slower killing as time goes. Also, in this spontaneous persistence, we have observed that there is a difference in the growth condition, so that if we grow the wild type E. coli in the glycerol, then we have seen the higher number of the persisters that are compared to the cells growing glucose, and the glycerol gives in this case, about twice as long doubling time than glucose. So there is also the growth rate condition dependence in this persisters. Another thing we also tried was to look at how the stress triggers persistence in this the exponentially growing dormancy, the exponentially growing population. So we applied the 1 hour downshift, and then we have seen the elevated level of the dormancy, and especially when we did it in the glucose growing bacteria, then we had quite large elevation of the persister level, while if we did this downshift in the glycerol growing population, then the level was less. So again, also indicating that the stress response itself does depends on the original growing condition. So, well, the overall, my impression is that it's not as simple as a bistable dormancy. And also that was also very clear in the Yin's talk, which talked about the depths of these dormancies. And this can come from the very different mechanism, and overall the mechanism seems to be very, very complex. But so there can be many answers to this, and there are of course the many fantastic experimental studies to dig into the mechanisms. But here I want to mention a couple of sort of the modeling thoughts, just to start thinking about what could happen in the cell and what could be a scenario to think that have this different dormancy and different responses to the growth conditions. So the first work I want to mention was done by the Yusuke Himeoka, who was a postdoc in our group. And what he got interested was if the sort of the basic carbon metabolism network have the embedded sort of the potential to cause the dormancy. And so what he did was he took the E. coli carbon metabolism core network, and then he put that on the mathematical model, and then simulated the dynamics. But very often when we simulate this kind of the dynamics, we set, say, the level of ATP as a constant, because that is it makes the treatment easier. But in his case, he actually treated this ATP as a proper cofactor that goes up and down depending on all the coupling reaction in the system. And then in the end, what we found out is that sometimes, depending on the where we start the initial condition, the cell can go either the growing steady state, but sometimes it's transiently go into the very low ATP state that is sort of the very dormant, and then after a while it comes back to the growth state. And it turned out that it's because there is a fertile cycle embedded in this the structure of the network. So this is one example where there is sort of the embedded possibility to transiently go into the dormancy when we just think about deterministic dynamics of the metabolism. Another model I want to quickly introduce is the one that Mikkel Food is the persister experiment proposed. And in this case, we thought about the interplay between the deterministic cellular growth and stochasticity caused by noise. I don't have time to explain the model in detail, but basically we tried to think of the simple allocation of proteins into the metabolic machinery that is responsible for nutrient and uptake, and the ribosome kind of class that is responsible for translation, converting nutrient into proteins, and then the kind of size class that is responsible for the division control. And then we put an allocation rule to each class so that the system allocate more to ribosomal class when there are enough nutrient. We then assume that these are the discrete unit to add the noise coming into the system and looked at the behavior. And then what we see is that if we invest the most of the fraction to this metabolic part, then the cells are pretty fine to grow exponentially. But if we limit the allocation to the M production to the relatively low level, then we sometimes see some different dormant behaviors. And then it turns out that in a sense that when we see the interdivision time distribution, then we saw the different peaks. Some are slower than the other. And in our model, this dormancy can be due to the lack of this metabolic class element or the lack of this ribosomal class element, or both. So in other words, in this model, we had sort of the different types of the dormancy. Sometimes they are because of the metabolic part became the bottleneck for the growth, and in other case the translational part became the bottleneck of the growth. So and we also could simulate a downshift of the effect of downshift in this case by setting the extracellular nutrient level for 0 for a while, and then we could measure how much the fraction of the dormant cells increased just after the downshift compared to the before. And then what we observed is that if the cells are growing at the high nutrient, then they actually have a stronger, get a stronger impact of this downshift, and get the higher level of the dormancy. And that is in short because when the downshift happens, these cells have a relatively high excess nutrients still left. And then in this case, we don't have the proper stress response regulation modeled. So the cells keep kind of dividing, and they have a chance to hit the state where especially the metabolic part of the component became the limiting to regrow again. So I don't mean to claim that this kind of models directly explained what is observed, but I hope that the thinking about these basic connections between the component and exploring what could happen can give us some ideas of how to understand the dormancy, and also this deterministic and stochastic, spontaneous and triggered, those sort of different categories of this dormancy or persistence are also all intertwined. So I hope that sort of this kind of the modeling give some starting point of discussion, including that maybe either you may not like it, and then you might have a better scenario to think about, and then I hope that will bring the field forward to discuss further. So thank you very much for your attention, and I'm looking forward to the further discussion.
Thank you so much, Namiko. That was really fantabulous and such a great complementation to some of the field and also molecular talks focus that we've been seeing so far. I'm going to move in now to our discussion. So I'd like to ask the panelists to please put on their videos so we can have a little bit of a discussion. We're going to try to extend this just by a few minutes so we can have a little bit of a discussion here before we wrap up the session. If you have questions for Namiko concerning her talk, feel free to put those in the Q&A, or questions generally for the panelists. We are monitoring that Q&A box, so let us know what you want to discuss. But I just want to start out with a few observations. And so something that I've noticed from the talks is that oftentimes when we're talking about measuring microbial inactivity or activity, it seems necessary to have this temporal component of the observation to be able to observe those dynamics. So for example, Catherine had her early spring versus late spring shift in which she was able to see kind of the reshuffling of who was active and then who became dominant at the next time shift. Ying had her different levels of response or different immediacy of response in the cells, in the persister cells. And so if she hadn't kind of looked over the course of within 5 hours, within this many hours, within, oh, they never woke up within the time frame we looked, she wouldn't have been able to observe those dynamics and follow up. And so that was one observation I made about the talk, is the importance of kind of like potentially looking at over time to be able to make a comparison. Another thing that I noticed across all the talks is kind of related to this, which is that the measurements we're using are quite different, though Rachel gets the prize for having the most different kinds of methods representing in one talk. That was pretty awesome. But also when we're measuring an indicator of activity or dormancy, we're measuring it not only with different methods, but at different targets. Like, for example, the agrosomes in those dynamics, are at essentially a single cell level. And then we're moving into a community level. We're looking at community level activity with metatranscriptomes and BondCat. And with spore enrichment, we're looking at particular populations. And so how do we bring all of these different methods and different foci of the methods, essentially different ways of measuring activity together to build some general insights here? So I don't know, does anyone want to provide some thoughts about that from the panel?
I mean, I can happily start to address some of this, I guess. I think what makes it hard to think about bringing all these methods together is that having a really operational one-size-fits-all definition for what dormancy actually is doesn't really exist yet. And based on what everyone's been saying, I'm not even sure it's really possible. So when you were asking about the methods, like, and in terms of just temporal activity, like my PhD student, Francesca, she's been looking at shifts in activity over 24-hour periods in the snow, right? So even on really short time frames, you have moments of activity, moments of inactivity. Some of it you might be able to capture with something like metatranscriptomics, but then if you need to do like a BondCat incubation, you might miss the window, right? And then some of the things you can do in a lab and other ones, you can do better in the field. So I think it's going to be really difficult to say, okay, this is what we need to do, this to help us define what activity versus dormancy actually is. So I think it's going to be tough.
Challenge accepted. Anyone? Any other thoughts about how we kind of bring together these different forms of dormancy? Ying, I'm curious, do you think that measuring agrosomes as an indication of dormancy depth, for example, in environmental communities is possible? Or do you think this is a technique that's good for cell populations that are growing in the laboratory and will be like completely impossible to translate to a more complex natural environment.
I think it's not so easy to using outside lab because the signature of the agrosome is just a forming foci. It's not a reduce of the protein. So we cannot detect the fluorescent difference between those markers. It's just a pattern difference. So I think maybe it's difficult, but I don't know, maybe in the future we can have some ideas on it. Thank you.
Yeah. Open to collaboration, it sounds like, everyone. So this brings me to another point, which is that when we think about dormancy and activity, we have a tendency to say, use it as a binary kind of category. Are you dormant or are you active? But what we've seen today is that there seems to be not only phenotypic heterogeneity in who's active or inactive, which we learned from Namiko, but also that there seems to be differences in intensity of activity, and even dormant cells are doing basic things like respiration. So how can we take advantage, I guess, of the methods we have, or what methods do we need to build or do we even need to do this? Is it necessary to move from this kind of simplistic are you dormant or are you not, to more of a gradient of activity and acknowledging the full gradient of biological activity that's possible? Is anyone interested in this kind of thing in their system? Rachel, you maybe you should take this one because you've got some data that could speak to this.
Yeah, I mean, that is one of the very difficult questions that we have in permafrost, because even cells that aren't known to form spores or persisters, they have such low levels of metabolic activity that in effect there is that they could be on that border of dormant. And so I don't have any great answers, but only to acknowledge that that is an important question, and I'm not sure how we end up addressing that. Especially because it's very hard to conduct time series trials when you're talking about ages that range into the tens of thousands and hundreds of thousands of years. I do like the advent of some of the newer technologies like BondCat FACS that at least don't require replication and just require low rates of protein synthesis. But it's I think it's a hard question that we're going to have to just chip at least at this point, with a lot of different methods.
Namiko, what do you think? Would it be possible to like more fully consider a gradient of activity rather than a binary?
I do think it's kind of necessary that we have to think about these different gradient, because it's very clear that the way to be a dormant there are many different ways to be a dormant, and each of them will have a different time scale.
Right. I think that was something really interesting about your talk, Namiko, is that you have these different responses depending on the condition of the cell. And it kind of brings me back to this idea that we were talking about in the first part of the webinar, which is like we were kind of philosophically talking about what is dormancy and what trade-offs are involved in a dormant state. But it seems like cells can be dormant for different reasons, as you said, and maybe trying to tease apart those different reasons and leveraging the methods that best address each deterministic trigger, that is, or even the stochastic ones, if possible, for entry and for dormancy. Because we've been talking a lot, especially in the persister literature, about dormancy as part of a response to low levels of nutrients. We think about this in the environment too, but you can also have dormancy as a stress response to a particular stress. You can also have dormant state assumed to enable dispersal, for example. And so those would be three very different reasons, if I can anthropomorphize a little bit, for cells to want to assume a dormant state. Catherine, in your system, you brought up the importance of dispersal, how you're having new cells come in and then contributing to the community. Do you feel like you can tease apart the influence of maybe activation or reactivation of those dispersed cells, given your time step analysis?
Yeah, actually we can, and we're looking at that now. And I think it's a question of timing. So for example, in these systems that kind of start over every year, right? The ones that get there first before maybe a stable community develops would probably be more sensitive to invasion from new organisms coming in from atmospheric deposition, right? But then as we get into the system where the communities are more stable, better established, where they're in more of like a competition type interaction system, then incoming organisms would probably be less successful. So we could look at this by, and this is something we've been doing, but I didn't have time to really look at or present, is we can look at what's in the atmosphere and then what's in the snow, right? And then follow that over time and see to what extent the atmosphere is successfully seeding the snowpack.
That's a good comment. That's cool. Maybe we'll have time to share those. You'll have time to share those data at another point. We have a question from Samuel Martins that I wanted to answer live here. The question is a hard one though. If we had to choose one measurement of activity and dormancy applicable to the widest variety of systems, what do you think it would be? Which is the best question to capture this idea of activity and inactivity?
Maybe I can answer.
Go for it, yeah.
Yeah, I think maybe bacterial single cell RNA-seq can be a very good tool to explore the heterogeneity in dormant cells, because as far as we know that persistent population is heterogeneity. It's not the same. Maybe one, and there are many types of persistent cells, and we are trying to explore its heterogeneity by single cell RNA-seq. And last year we have published an ELIFE paper on the new technology, and we are also working on it for exploring bacteria such as tuberculosis, something like that. So I think it's a quite powerful technology to explore the dormancy continuum. Yeah, thank you.
Single cell RNA-seq. That's where we need to go. Measuring the heterogeneity in mRNA at the single cell level and being able to relate it at the population level. Do the rest of you agree? Do you have a different method that you would recommend that you think would be most broadly generalizable and comparable across different systems?
In an ideal world,
sure.
And also
we're dreaming.
Yes, and also when you have, what is the phrase? When you have a hammer, everything's a nail. And so, we sequence a lot of stuff in my lab. I would give a whole lot to be able to do transcriptomics from a lot of these systems, because we just know so little about it. I also would love to see bond-cat-fax develop to the point where it is a regularly usable resource for that. But that's, of course, looking at kind of large, complex communities rather than the kind of finer resolution view that single cells will give. So for my system, for my type of system, those are the tools that I would love to see be able to work consistently in these challenging environments.
Good point. Going from the lab to the environment and back again presents some challenges. Okay. I see that we're now officially over time, so we should probably wrap up in the next minutes. I just want to check in with the panelists. Is there anything else that you'd like to elevate or, just kind of highlight from your thoughts of the session? We've had such diverse talks, to environmental and to more lab and theoretical, clinical, bugs, E. coli, and all the way to like bacteria, Clostridia living in permafrost. So we've really spanned a breadth of, I think, environments and methods here. Closing thoughts. Namiko, can I start with you?
Yeah, it's sort of fascinating to see this diversity, and especially in the environment. And it's great question that that is a method, and what is the sort of good definition of the dormancy. But it's probably difficult to get one definition. But I think it's great to keep questioning: is our method picking everything we want to know, or are we only seeing this because we do only this method, and then try to expand what could else happen? That I think is a very important part of this kind of research.
Yeah, I agree. That's an excellent thought. Rachel, moving over to you. Final comments?
This is, I guess, bringing this to a gap in what I see in terms of the literature. So this is a little bit specific because we've kind of talked about big picture, but I would love to see more attention focused on the microeukaryotes and activity and dormancy, because they are potentially crucially important for especially systems where anthropogenic change is threatening. Anthropogenic change is influencing the carbon cycle in those systems, and so may contribute to climate change. And understanding the activity and role of those microeukaryotes in those systems, I think, has very large consequences for global change. So that's a little bit specific, something that I'd love to see more of, and something that we weren't even quite aware of until we saw this enormous abundance of fungal and protist RNA in our thawing samples.
Kind of integrating measurements of active and inactive, not just focusing on the bacteria and archaea, but also looking up towards towards the larger organisms as well,
Right.
and like trying to make
Right.
conclusions using a more
Right.
food chain or holistic perspective.
Right.
Okay.
And
Yeah.
these definitely form very substantial dormant states in these systems as well.
Yeah. Yeah, they may be overlooked. That's a great comment, Rachel. Thank you. Ying Ying, do you have any final comments before we close?
Thank you very much for this great session and the community to share our findings. And I hope in the future we also can have the opportunity to share our findings in the future. Thank you.
Thank you. Catherine, we started with you. Let's end with you too. Do you have any final comments or observations you want to elevate about dormancy and measuring it in different systems?
Yeah, I think I just want to say, like, to start off, it's such a fun thing to think about because it's complicated, and all of us work with things that we don't actually see with our eyes. So, we have to take risks with how we look at things. And I think the new technologies are going to help us get a better handle on when something is dormant and when it's not. I think what would be useful and interesting is maybe some of the stuff that you're actually doing, Ashley, where you look at different extremes of dormancy and maybe classifying this continuum of dormancy relative to how easy it is to resuscitate or get the organisms out of dormancy might also help us like better define what dormancy is under which conditions, and also maybe help us find tools to look at those different states. So not a real maybe more that, yeah, the research is moving forward. And I think like also having these moments where we can talk to people who are looking at this using different systems and approaches is really useful for all types of research, be it in the lab or in the environment. So yeah.
Yeah, that's a great comment. We actually had a Q&A comment that was asking about how to reactivate cells from dormancy or that are viable but non-culturable so that we can cultivate them. And I didn't bring it up to answer live because I felt like we didn't have an answer collectively, but but I see that as an interesting way to think about dormancy, is like who can we reactivate, right? Under what conditions can things reactivate? So that could be an interesting future direction as well. Okay. Well, thank you everyone for your engagement and participation and for sharing your perspectives on how we measure and think about microbial dormancy. Thanks to all of the attendees for your questions. Feel free to check out the part 1 on recording of the webinar on YouTube. The recording of this one will be available, but not immediately. It will be a couple days, so please be patient. And then also, we welcome any submissions to the special collection of M systems on dormancy. If you're working on topics related to this area, please feel welcome. And finally, if you're going to be attending the ASM Microbe meeting in Los Angeles this summer, we have a symposium on dormancy that's going to be there, and we welcome you to come join the symposium and continue this, I think, really important discussion for understanding microbial systems better and thinking about how we can manage them and understand them. So thank you, everyone. I wish you a great rest of your day. And yeah, this is super. Thank you so much.